Welcome to the Pancreatic Cancer Action Network's webinar, Immunotherapy for Pancreatic Cancer. My name is Cassidy Moravec, and I am the Senior Manager of Clinical Initiatives for the organization. At the Pancreatic Cancer Action Network, our goal is to provide you with better resources so that you can make informed decisions. Patient Central Associates can provide you with personalized information free of charge on topics such as diet, nutrition, and treatment options. Contact Patient Central at 877-272-6226 or email at patientcentral@pancan.org. I would like to take this opportunity to thank our webinar sponsor, Lilly Oncology. And we would also like to thank our dedicated partners in progress listed on the next slides. The webinar is scheduled for 60 minutes and includes a presentation followed by a Q&A session. A recording of this presentation and the slides will be available under the Educational Events page at pancan.org. Now I would like to introduce our presenter. Dr. Robert Vonderheide is a medical oncologist, the Associate Director for Translational Research, and the co-leader of the Immunobiology Program at the Abramson Cancer Center. He is Vice Chief of Research, Hematology Oncology Division, and is the Hannah Wise Professor in Cancer Research at the Perelman School of Medicine at the University of Pennsylvania. Dr. Vonderheide is also a member of the Scientific and Medical Advisory Board for the Pancreatic Cancer Action Network. His laboratory combines efforts in both basic research and clinical investigation to advance the understanding of tumor immunology and to develop novel immunotherapies for cancer. Dr. Vonderheide, I will turn the presentation over to you.
Well, thank you, Cassidy. Welcome everybody to this webinar. I'm going to discuss immunotherapy for patients with pancreatic cancer today and speak for about 45 minutes or so and then break for a question and answer session. I'm going to get started there. So the excitement in the field of cancer at the moment is the dawning of a new age of cancer immunotherapy for treating patients with cancer. And that has really created a great deal of excitement because it has provided us with a handle on a new modality to treat cancer. And this slide shows some of the covers that appeared on scientific literature announcing this new breakthrough and indicating some of the modalities that I'll touch upon today. What I'll do first is review some of these new developments over the last few years in general, and then talk more specifically about how we're going to make this happen for patients with pancreatic cancer, and finally what our progress has been. So for many years, the notion of cancer immunotherapy was speculated about. It actually is an idea that dates back more than 100 years. But it's really been in the last few years that we've begun to see the future being now. And we've seen the development of novel antibodies that really act by cutting the brakes on the immune system, and I'll talk about that. There's also a great deal of effort in genetically reprogramming our patients' own immune cells so that they attack cancer upon reinfusion, and sometimes one version of those cells is called the CAR T cell, and I'll mention that in more detail. And in other aspects, in the last year have been new efforts to use genetically engineered viruses that upon injection into a patient's tumor, shrink that tumor by activating the immune system. I must say there are many, many more experimental therapies coming and that we'll see roll out over the next while. In 2016, this slide summarizes where we're at. What we've been able to see from immune therapies is that a subset of patients really do clinically respond, and that this has been across a variety of effective approaches. And many of you will know that many of these approaches are now FDA approved. Certain antibodies that cut the brakes 3 of them have been approved over the last several years in many indications, ranging from patients with melanoma, lung cancer, kidney cancer, Hodgkin's disease, and probably many more approvals coming. And I'll spend some time about what those therapies really are. But regardless of what type of immune therapy is being looked at, for patients in whom a response occurs, in other words, for those patients where the tumor really does regress, what we're observing with immune therapy is that the response can be quite durable, years and years. And we've seen patients go into complete remissions, and those remissions last for many, many years. There are some toxicities. These toxicities are different than we observe with chemotherapy or radiation therapy, and they tend to be toxicities associated with the immune system. They're immune-related, and they have everything to do with the fact that we have activated the immune system, and instead of attacking cancer, the immune system can sometimes attack normal tissue. But in general, particularly as more time passes and there's more experience using these drugs, the immune toxicities tend to be manageable, and they're even reversible. And as I said, they're quite a bit different than what we're used to. There's none of the bone marrow suppression or nausea, vomiting, hair loss that we see with conventional chemotherapy. And we're at the point now in the field where we're beginning to see early reports, promising reports, with many of these different types of modalities being combined. So that's a very important thing that's happening. This effort has caught the national attention. Probably no better demonstrated than the cancer Moonshot, which is devised to cure cancer. This is the effort led by Vice President Biden. Vice President Biden launched this Moonshot at my own institution in January of this year when he visited, shown here in this slide, visiting our laboratory where we generate engineered T cells. This is Bruce Levine, who runs this laboratory. This is Carl June, the overall investigator. This is Amy Gutman, who is the president of the University of Pennsylvania. And we had the opportunity, many of us sitting here in white coats at the University of Pennsylvania, to meet with the vice president and the directors of both the NIH and the NCI, shown here, and discuss the future of immune therapy and how it fits in with the overall effort, new effort to attack cancer on many levels. And we feel certain that immune therapy will be a central plank for this effort going forward. Other efforts that have gained national attention have included the launching of the Parker Institute for Cancer Immunotherapy. This is a really unprecedented six-center effort led by Sean Parker, who's shown here, who is a leading businessman and technologist who decided to establish with his own funds a very large institute aimed directly at cancer immunotherapy. And what's shown here in this circle are the 6 founding institutions, including my own and many others. And this institution is dedicated toward bringing cutting-edge immune therapies forward. And there is a high priority to study and work on pancreatic cancer through this institute as well. But the downside and what we must realize for those of us concerned about pancreatic cancer is that most patients, and especially those with pancreatic cancer, do not as yet respond to current immune therapies, that some of which are now FDA approved. So we need to redouble our efforts to make this new type of therapy effective for everybody, including those patients with pancreatic cancer. This slide summarizes why pancreatic cancer is been so difficult to attack with any type of therapy, including immune therapy. And there are clinical and biological challenges that face us, and all of us on this phone call know all too well. Early detection in pancreatic cancer is difficult, and there are some great new ideas coming forward, but as yet we do not have an early detection marker that is reliable and effective. there is a suboptimal clinical response to standard therapies, including chemotherapy and radiation therapy. And although we've seen the advent of combination chemotherapies over the last few years, which no doubt has brought great benefit to many patients, it is still the minority of patients that respond to chemotherapy, and we need to address that. The main tumor mutation in pancreatic cancer has to do with this molecule KRAS. And unlike other mutations in other tumor types, so far we're finding that KRAS is currently undruggable, meaning there's no clear drug that can block the action of this mutant KRAS, which is so much a part of pancreatic cancer cells growing, and many other types of cancers. This is actually another national effort that was launched several years ago, an initiative to study the KRAS oncogene and to find out, is there a more creative way that we have in our midst to address KRAS. We're very much hopeful about this opportunity because we feel that in addition to the ways mutant KRAS drives tumor cell growth, we feel that it also at some level orchestrates suppressive activity against the immune system and poses a big problem for any immune therapy that we don't see in many other tumors. And if we can derail KRAS, we think that immune therapies may be easier to come forward. And finally, in large part due to the KRAS oncogene, but many other mechanisms, there's a hostile microenvironment in pancreatic cancer, meaning where the tumor cells sit and I'll show you a photo, is a very active amount of scar tissue and other type of inflammation. And that inflammation, again, is counterproductive to the types of immune cells we want to attack pancreatic cancer. So we need to rethink the problem and understand that pancreatic cancer is not the same as melanoma. Melanoma, fortunately, is the disease where these immune therapies first took hold, and amazing results are being shown and brought forward for patients who needed them, because up until 5 years ago, there was really nothing much to offer patients with melanoma. Certainly chemotherapy was highly unreliable. And it does give us hope to know that there was such a tumor years ago, the kind we're facing now, looking at pancreatic cancer, where we were able to finally bring immune therapies. So we can build on these prior successes, I think, but we won't be able to just cut and paste from the FDA-approved drugs that we have and expect that they will work for our patients with pancreatic cancer. And what we need, and in fact we are gaining day after day, is a deeper understanding of the underlying biology, especially the surrounding tissue, the surrounding stroma, the actual non-cancer part of the tumor that restrains the immune system and holds it in check in pancreatic cancer. So this is a slide to illustrate what we're talking about with regard to the immune biology of pancreatic carcinoma. So tumors develop in the face of a mutation in KRAS and also the loss of tumor suppressors, TS, and it creates this picture which is quite notorious. This is a picture of pancreatic cancer under the microscope, stained with this red, white, and blue stain. These are the cells of pancreatic cancer shown here. And what you can see is just a small part of this picture is actually the pancreatic cancer cells. The rest of it includes all this material out here. These other small blue cells, which represent inflammation and fibrosis, shown here in this pink staining. And these cells, cancer cells, sit in this nest and hide themselves, make it very difficult for us to treat. And we call this the desmoplastic stroma, which acts somewhat like a barrier. And in fact, we know that conventional chemotherapy, such as gemcitabine, which we've used for more than 20 years in this for patients with this disease, has a hard time penetrating this stroma, and it means it's one of the reasons why gemcitabine is only partially, marginally effective in this cancer. The other cells and the other aspect of this tumor include fibroblasts, macrophages, B cells, other parts of this matrix, which is this pink stuff, immature myeloid cells, regulatory T cells. These are all inflammatory cells, which are not the ones that generally we think of as having any capability of shrinking the tumor, but rather they suppress the other parts of the immune system. And what we generally do not see in this tumor is infiltration of so-called effector T cells. This is the cell that we're trying to mobilize with immune therapy. This is what the checkpoint blockade antibodies do and what the engineered T cells do. But you can see that in the tumor microenvironment of most patients, there are very few of these cells. And so bear with me as I go through this biological description. What we understand now is that tumors arise, any type of tumor, and in many cases, a T cell response is made, and that leads to the elimination of this tumor. The tumor, in certain types of diseases, has no choice but to edit itself, sort of like under the pressure of our Darwinian-like pressure. The tumor will resculpt itself and escape, and it's a very hard tumor to treat with immune therapies. Another aspect of this is that in the face of this T cell response, there's an anti-response made. Both the host, ourselves, and the tumor organize immune suppression and really block out these T cells and create what we call peripheral tolerance, and that also leads to escape. Now, one of the amazing discoveries over the last 10 years has been that this mechanism is driven by very specific molecules, sometimes on the surface of those cells that I showed you. And they have names like CTLA-4 and PD-1 that you might be familiar with if you're interested in immunotherapy. And these monoclonal antibodies, or both, can be used to treat a tumor as long as there's a pre-existing T-cell response. So this slide illustrates two types of therapies that have really taken center stage for treating cancer. The first that was FDA approved, is an antibody that blocks CTLA-4. And this is a picture of how it's supposed to work. These are the antibodies, these Y-shaped molecules. And when given to patients by vein, they find themselves into the deep tissues and identify their ligand, what they bind to. And it's this molecule, CTLA-4. As you can see, this molecule is on the surface of a T lymphocyte. This molecule serves as a brake on the activity of this T cell. All these other molecules I'm showing here, I won't go through detail, are receiving signals from other cells in the immune system to activate. So the T cell will activate when seeing these signals. But if this molecule gets in the way, the whole T cell turns itself off. Well, if you give an antibody to this T cell and you eliminate that, the T cell turns back on again. And something very similar was the development of two other agents, and there's now several of these compounds which are FDA approved. And they bind to molecules such as PD-1 or PDL-1. Ordinarily, PD-1 and PDL-1 bind to each other, and again, this is a way to turn off the T cell. Even in the face of this ghost signal shown here, either the tumor or another cell in the immune system can express this molecule PDL-1. It binds to PD-1, and that's a break. But if you can give a patient anti-PD-1, these PD-1 antibodies, or PDL-1, either one, it can interrupt this activity and stop the negative immune signaling. And these things these antibodies have been shown to be highly effective. Again, not in all patients, but in patients with certain types of cancer, and we're even beginning to combine them together to see even more potent responses when the two are used together, because CTLA-4 and PD-1 are completely different pathways. What this means is that if you give these antibodies, you can activate T cells that pre-existed and that were somehow dysfunctional, or sometimes we call them exhausted, and these T cells gain their activity again and go back to killing the cancer just as they were built to do. And what we're beginning to understand is what kind of patients respond. And listed here are any number of features of a person's cancer that can predict whether or not they'll respond to these antibodies, which are collectively known as checkpoint blockade. And you can see some of them have to do with, are there pre-existing T cells in the tumor? Does that tumor express PD-L1, and so on and so forth. One point I would make, and it's a real point of exceptional hope for our patients with pancreatic cancer, is that a rare type of inherited cancer, which has to do with how DNA is maintained, and sometimes known as microsatellite unstable, or MSI high. These types of tumors, which again run in families, can involve not only colon cancer and endometrial cancer, but every now and then, about 1 or 2% of the time, they can be involved in pancreatic cancer. And so recently, a few, not more than a handful of patients with pancreatic cancer, on the basis of MSI high, received anti-PD-1 antibodies, and in reports recently given, the majority of patients with this type of pancreatic cancer do actually respond. Now, it's just a 1 or 2%. But I pause on this because this is an amazing opportunity, and really goes into the category of a stone not to avoid looking under. We recommend that every patient with pancreatic cancer now be tested for whether or not they're MSI high, in the 1 to 2% chance hope that it is in fact that's the case, and a number of centers have the ability to do that testing. What it means in big picture is that these are patients with pancreatic cancer whose tumor has all those same stromal and desmoplastic features, and yet they are responding to immune therapy. This example, this type of pancreatic cancer, tells us that immune therapy can be highly effective in this disease, and we shouldn't give up hope. But one of the aspects in most patients with pancreatic cancer is that, in fact, the immunosuppression happens first, and that there's a very weak T-cell response that happens inside the tumor, creating what we call an immune privilege, so that the tumor doesn't have to expend energy escaping. It just sits there in this shroud of tissue and hides from the immune system. And what we're learning is that this really needs to be overcome by a combination of vaccines and blocking those immune checkpoints. And this will be the theme that I'll really emphasize as we go through the talk. Now, immunologists have a reputation of being very difficult to understand. I hope that's not the case today. But we call tumors that have these T cells in the middle of the tumor hot, and tumors that are lacking such a T cell response cold. So hot tumors and cold tumors. And what we know is that melanoma can very often be a hot tumor, and this is a stain of a patient with melanoma, and all these brown cells are not actually tumor cells, but they're T cells in the middle of the tumor. And this patient with this tumor, given CTLA-4 or maybe PD-1 or both, will respond, have a high likelihood of responding and shrinking that tumor. But a very typical picture of pancreas carcinoma is that being a cold tumor, where we see a single T cell in the middle of this tumor. And we know now from a number of patients treated, with the exception of patients who are MSI high, that the response to CTLA-4 or PD-1 or both is exceedingly rare in patients with pancreatic cancer. And so we're looking to understand how can we convert this cold tumor to a hot tumor and make them now responsive. This just is a little piece of laboratory data from my group to indicate that this is possible. This is a laboratory model of pancreatic cancer, and you can see that when we look for T cells in the middle of this tumor, we again see very few. And if we use PD-1 and CTLA-4, or the combination, to try to treat this tumor, we see that there is really no tumor response. But we noted that if gemcitabine and nab-paclitaxel, which are two drugs now used quite commonly for patients with pancreatic cancer, are combined with another immune therapy I'll talk more about later this hour, and use an agonistic CD40 antibody, you can see now lots of T cells in that tumor, the brown cells, and if you then treat with PD-1 or CTLA-4 or both, we see regression and even cures in this laboratory model. So this, again, is giving us great hope. And so what we see for patients with pancreatic cancer is that it's not enough simply to turn off these negative factors on this teeter-totter, but we actually have to turn on the T cells, because by and large they're missing. So we need to do both of these. If we just step on the gas with a vaccine, we don't expect any response. If we just give checkpoint blockade, we don't expect to see and have not seen responses. But if we combine both a vaccine of some sort and checkpoint blockade, perhaps we will begin to see tumor regressions. Now, unfortunately, over the last month, 2 large studies involving novel vaccines for patients with pancreatic cancer both reached maturity, and the results were reported, and both the vaccines turned out to not be effective in randomized studies for patients with pancreatic cancer. And this was at once quite disappointing, of course, for all our patients. But on the other way of thinking, some of us were not altogether surprised that the vaccine alone was not able to accomplish its goals, and that we know that these vaccines are actually effective in turning on T cells, but we need to combine them and use them in combination. So this slide summarizes the opportunity for combinations, and this is how we're going to break the ceiling of what single or double checkpoint blockade can achieve. So here are a variety of strategies of all the things we think we need to target and work on, and a great deal of work is happening in this regard. And I don't want to leave the impression that we're without reagents or without ideas. This slide is summarizing all the many drugs. Everything listed here represents a novel pharmaceutical agent that's being developed. Nearly all of these are already in clinical trials, and we're beginning to understand how to use them, because we think that there are checkpoints beyond the 2 I mentioned. Particularly in pancreatic cancer, we have evidence to think that some of these other ones may be actually even more important. We need to generate T cells, not only using vaccines, but other types of modality, radiotherapy, which I'll talk about, chemotherapy, as I mentioned previously, and these CAR T cells. And then we need to understand that we're dealing with a very hostile microenvironment and try to alter it. And it turns out we know and have discovered as a field many switches that will convert this. Many of these things are shown here. The I means inhibitor, so these pathways being blocked. And again, a lot of these are in clinical trials. Once we generate the T cells, we need to get them into the tumor, and there's great ideas for that. How the T cells come about, there's additional mechanisms, and how to actually give the T cells gasoline, so-called agonists. There's any number of therapies. And as I said, all of these are coming and already in clinical trials. There's a great challenge, though, with trying to figure out combinations. When you have to use more than one therapy, how do you know which ones to use? And there are really very many pathways and many drugs that it's not just a matter of putting together the few combinations we can think of, but how are we going to prioritize the many, many combinations? I would say one of the difficulties is that there are too few patients with pancreatic cancer on clinical trials. The national average is only 5%. That means that 95% of all patients with pancreatic cancer receive the same therapy that we've had for years, and we know the outcome of that. And we really think a huge opportunity is for patients to join clinical trials, for organizations to run more aggressive and state-of-the-art clinical trials, particularly as we have this opportunity to test novel immune therapies. We need to recognize that there may be a diminishing marginal return, not to mention toxicity, if we continue simply to use checkpoint inhibitors and not to take full advantage of all the knowledge that we have in immunology. And this is a question we all need to think about as we go forward, is that these drugs right now are extremely expensive. It's not the case that even though the drugs are FDA approved, that one can simply easily buy them. They can cost $100,000 for a year worth of therapy. So how will we be able to afford it? And if they remain this expensive, we need to pick the very best ones for patients. And another critical need is that we need to understand which patients will respond to which therapies. This is a notion that's really heart and center of the Vice President's initiative on the cancer Moonshot, and that is to revolutionize personalized medicine with regard to the immune system. And we need to understand much more about our patients' tumors than simply what we're doing now, which is, for the most part in the field, simply testing if they express that one molecule. What we really need is a comprehensive profiling effort, which involves understanding the DNA, the RNA of the tumor, the surrounding and elaborated proteins, the metabolism. And all these types of assays can be performed and are routinely performed, but We have not yet integrated that knowledge with what we need to do and the types of other immune therapies. What we have established are very large means in this country and at many medical centers to sequence patients' tumors and understand the mutation. And there's a great effort to do that. But the early data from doing that for patients with pancreatic cancer is that we're identifying in the vast majority of patients only a few mutations, such as the KRAS mutation. And so far, these mutations don't carry with them an obvious drug to use. There are exceptions, and there's great hope here. Patients who have an inherited mutated copy of BRCA1 and 2, those individuals are at risk for pancreatic cancer, and patients who develop pancreatic cancer on that mutational basis, there is a collection of new drugs, one of which is already FDA approved, called PARP inhibitors, which is showing great promise. But again, this appears to apply to about maybe as many as 5% of patients with pancreatic cancer. It's a low number, but it's an opportunity worth exploring, which is why here at our center we are very aggressive about understanding whether a patient before us has an inherited mutation in BRCA1 and 2, because the therapeutic opportunity is very much real. But in terms of immune therapy, our goal is to identify a biomarker that will predict which patients respond to which therapies, and so that we only treat those patients most likely to respond. We eliminate risk of these therapies by not treating patients who we know will not respond. And this is a way to address what some people call the financial toxicity of immune therapy by really focusing on drugs that provide value for the money and help patients. So in the last little bit of this webinar, I wanted to go through a few examples, not by any way with the impression that this is a comprehensive list of everything that's out there, because there's a great number of efforts to bring forward not just immune therapies, but other types of therapies for patients with pancreatic cancer. It's really quite different than it was even a few years ago. But with regard to immune therapy, I want to highlight a few national efforts. One that I'm involved with, which is a partnership with Stand Up to Cancer and the Lustgarten Foundation, which is a national effort of 7 institutions listed here to develop next-generation immune therapies for pancreatic cancer. And you can see them listed here. A vaccine, this is a vaccine with checkpoint. The CD40 antibody plus chemotherapy, this clinical trial very much based on the laboratory data I showed you earlier today. Chemotherapy plus an inhibitor of a certain kinase, and other types of antibodies and inhibitors that activate the immune system. Let me just pause and tell you about one clinical trial, because this really summarizes what we're trying to do. So this is a clinical trial. It's a national clinical trial. It's actually nearing its enrollment of combining a vaccine, which is 2 types of vaccines, something called GVAX and a type of vaccine built out of a bacteria called Listeria, in combination with a checkpoint blockade. So this is vaccine plus checkpoint blockade. Here's the GVAX vaccine shown here. It's a whole tumor cell vaccine where patients get a genetically modified and lethally irradiated tumor cell, followed by an infusion of attenuated Listeria monocytogenes, which is recombinant, meaning that it's been engineered to express this pancreatic cancer target, mesothelin. And in that setting, these two work well together to generate T-cell responses. And the clinical trial is now going forward to combine that vaccine with nivolumab, which is an antibody against PD-1, to, again, derail the immune suppressive aspect. Here's how the study is working. It's a randomized phase 2 study, again, through our Stand Up to Cancer team, where patients are randomized to either get the vaccine and anti-PD-1 or the vaccine alone. Along the way today, I'll point out these NCTeen numbers, which you can find on the web simply by searching that number, and you can get a lot more detail about this trial. Now, this trial is nearly enrolled. We were able to enroll more than 100 patients in a matter of a few weeks. So hopefully the results are positive and we can go to the next step. Another idea, and again, involving this molecule CD40, has to do with a whole nother way of using the immune system to destroy pancreatic cancer. This is an effort, very pleased to say that the Pancreatic Cancer Action Network was among the first to fund this study out of our laboratory. And this is an antibody called CD40, which enables immune activation in a completely different way than the PD-1 or CTLA-4 antibodies. And what we notice in the laboratory is that if this is pancreatic cancer with this network of stroma shown here in blue, that following anti-CD40, we were able to destroy that network and really expose the tumor cells there shown in red for the immune system to attack. This enabled us to treat patients with an antibody in combination with gemcitabine chemotherapy, and these are CAT scans from a patient on that clinical trial that showed major regressions of this tumor in the pancreatic head. This is the pancreas, and as well as these liver metastases disappearing. This type of notion of CD40 with chemotherapy has prompted a new clinical trial, and here again is the NCTeen number at the University of Pennsylvania and Johns Hopkins Medical Center, where patients receive a chemotherapy, gemcitabine and Abraxane, along with this anti-CD40 antibody, which has this R0 number now. This clinical trial is for patients who have resectable pancreatic cancer, not for patients with locally advanced or metastatic disease. We hope to eventually open through the Parker Institute a similar study for patients with advanced disease, but at the moment this is the study that's open. Another strategy I'll mention briefly is using radiation therapy, one of our oldest therapies, but using it in a completely different way. And again, in the laboratory, what we were able to find is that radiation given to one tumor can make another tumor elsewhere in the body shrink, and that this only occurs when the radiation is given with immune therapy, checkpoint blockade. And so far what we found is that this strategy, which we call RADVAX, can generate a vaccine that is functional in just about any cancer we've tested, including pancreas cancer, actually. And this effect occurs because of T-cell activation. And what we can see is that the best results occur if we use radiation in a novel way, rather than giving it every day for several weeks, but rather use it all at once or in a short course over a few days. This is how it's supposed to work. This is a patient with tumors shown here in the blue. If we radiate this one lesion, say this lesion in the lung, and follow that with these antibodies against certain checkpoint blockade, we can certainly make that tumor shrink. But what we find is that the immune system spreads because it goes everywhere in the body and can shrink those other tumors. And so we hope that this is a new modality. We've seen some success already. Again, this is a patient with melanoma where we radiated this lesion on this patient's flank shown here in yellow, and it goes away with radiation therapy. But other tumors far away from where the radiation were given also shrink. And this has enabled us and many others across the country to look at combination therapy using radiation and checkpoint blockade. Here are two clinical trials at the University of Pennsylvania, but there are many others across the country. I'll make a final point about the power of the immune system before we turn to the Q&A, and that is something that the founder of the University of Pennsylvania, Ben Franklin, said more than 300 years ago, and that was, an ounce of prevention is worth a pound of cure. And we have to stop and pause as immunologists and realize that far before the use of cancer immunotherapy, using the immune system to treat cancer, the last revolution using the immune system was really childhood vaccines. And the key about those vaccines was that we have to vaccinate children before they get the disease. The same holds true for the use of flu vaccination today. You need to get your shot before you actually have the flu. It doesn't work once the disease takes hold. And so we think that now, having observed these great regressions that can occur in patients who have cancers that we can see, maybe now we're beginning to understand the knowledge of how to activate T cells, for example, in individuals who are at risk for cancer, and to prevent cancer from forming in the first place. And I think this is the next great challenge for us, to convert our new knowledge and our new success with immune therapy into practical and effective means of immune prevention. Certainly, there's already great examples. Vaccination against certain viruses like HPV can eliminate infections of that virus in individuals who get vaccinated. And because those viruses are related to cancer, we can impact certain cancers that are related to viruses. So we know on many levels that this approach can work. And I'll just highlight to you a final clinical trial, which is shown here again, the NCTeen number. And this is a phase 1 study where we're using gene therapy. We're using a DNA technology to combine a target, a bullseye, along with an immune molecule for activation. And the bullseye that we're using is something called TERT, stands for telomerase reverse transcriptase. And this molecule is overexpressed in more than 95% of all human cancers, including nearly every case of pancreatic cancer. And this molecule has to do with helping the tumor cell keep its chromosomes intact and not falling apart. And so this enzyme is reactivated in cancer cells nearly universally. IL-12 as a DNA vaccine has been used in the infectious disease community for a long time. It's not yet FDA approved, but it's the experience in the development of vaccines for HIV have been most promising. And so, partnering with Inovio Corporation, a vaccine was developed combining this target as DNA and this cytokine as DNA, all synthetically made. And this DNA is placed into this syringe as a vaccine. So this clinical trial is happening now. In the first case, eligible patients have pancreatic cancer or other types of cancer, but these are patients in remission after their surgery. So, for example, after the Whipple, and after patients have completed their adjuvant therapy, whether that's chemotherapy or chemotherapy in combination with radiotherapy. Once patients complete that, we know that unfortunately the relapse rate in patients with pancreatic cancer can exceed 75%. So in order to try to use the immune system to prevent that relapse, patients are being enrolled on this clinical trial where they'll receive this shot shown here every month, and that's done 4 times. And it's applied through this device, which actually sends electricity at the point in the muscle where the vaccine is given using this device called the Selectra. That notion of DNA with electroporation has shown great promise in other medical settings, and so is now being carried forward. And of course, because it's a phase 1 study, we're looking to make sure that it's absolutely safe. And secondarily, can we generate an immune response that will prevent the cancer from relapsing? If this actually turns out to be safe, and so far it is quite safe, and the immune response is robust, then there is a hypothesis that we could bring this vaccine forward for patients who are at high risk for developing cancer, but are actually not patients. They're healthy individuals. One example would be to use this type of vaccine or others that could be created along this notion for patients who have mutations in BRCA1 or BRCA2 who are at risk for developing pancreatic cancer. And if we could show that that was effective, then there could be other types of individuals at risk for pancreatic cancer who might one day benefit from such a prevention vaccine. This is a big idea and a long way off, but it's important to note that the state-of-the-art of the immunology is so sophisticated as to enable this type of clinical trial and a great deal of effort going forward. So before we break and moderate questions, I did want to just make a few conclusions. I do think there is a road to success using immune therapy in this disease, and that I can't understate the massive efforts nationwide and across the world in pancreatic cancer research. This wasn't always the case, and I must say that organizations like the Pancreatic Cancer Action Network have really inspired the nation to take a hard look at this problem, fund the research necessary, train the individuals, and teach investigators about big demands and the biological challenges of pancreatic cancer, and it's just been overwhelming over the last 5 years to see a true, robust community of investigators, clinicians, combined with basic scientists, really targeting success for our patients with pancreatic cancer. And these new discoveries are not happening in a vacuum. They're really driving novel therapies. I would dare say that not a single one of the clinical trials that I showed you today could have been possible even 5 years ago, because we didn't have the knowledge of the tumor, we didn't have the knowledge of the immune system and how to go about it. And there's a great deal of effort in pancreatic cancer, as I've outlined today, to develop novel therapies. And finally, the clinical trials are absolutely critical, as I alluded to earlier in the presentation. This is a way, not the only way, but certainly a great way of bringing tomorrow's therapies forward for today's use, and to make them possible, to study them, to optimize them, and to prioritize the ones that we feel will work the best. So at that point, I do appreciate your attention, and we'll begin the question and answer portion.
Thank you, Dr. Vonderheide. We will now begin the question and answer portion of the presentation. The first question for you, Dr. Vonderheide, touches on clinical trials. You touched on them briefly throughout your presentation. So the question is, will collaborative immune therapy clinical trials be available soon to regional cancer centers around the United States, and particularly in remote areas?
Yeah, this is a great question. And we're struggling with that, because most of the clinical trials that we're talking about for pancreatic cancer are phase 1, and those types of studies tend to happen at the tertiary care hospitals and major medical centers. And a recent study by the Pancreatic Cancer Action Network identified geography as one of the biggest stumbling blocks. As it turns out, many of the major cancer centers, such as ours, has a community network of regional hospitals, and we've begun to partner with our regional network to enable bringing clinical trials to everybody and making them more accessible. One of our regional hospitals has actually developed a concierge transportation service to bring patients close to the major medical center for the phase 1 studies. The more rapidly we can do the phase 1 studies and develop phase 2 and phase 3 studies, which are more amenable for patients who are in the community away from the major medical centers, the better. And that's where we hope to move things as fast as possible.
Great, thank you. And our next question is about the timing of immunotherapy. So at what time after diagnosis is immunotherapy best given for the most possibly effective treatment?
Yeah, this is also a great question, and one of the things I think we have to recognize is that the immune therapies we have at the moment are not proven enough, and certainly they don't work fast enough for patients who are diagnosed initially. Say a patient who is initially diagnosed with pancreatic cancer, with metastatic disease. So we don't think at that point in 2016 an immune therapy is the best way to go. But rather, we're looking at two options. One is to treat those patients with chemotherapy and stop the disease from growing, get it into the best remission we can, and then follow that with immune therapies. One of the vaccine trials, the GVAC study I showed you today, has that exact design. And other approaches that we're using are to simply mix standard therapies such as radiation therapy and chemotherapy with immune therapy at the same time, have it in there in the mix. And we have to balance the fact that chemotherapy can damage the immune system sometimes. But the more we dig into this, the more we realize that certain immune therapies actually, if used not in certain ways, can actually enhance the immune system. And so there's many thoughts to combine standard therapy with immune therapy all at the same time. I think this was, we're borrowing some of this thinking from our colleagues who are developing therapies for lung cancer, for example. But as they've made headway, they're realizing that if they get the right immune therapy, sometimes it works better than chemotherapy. And that's really ultimately the goal, is to bring immune therapy forward where we can rely on it to work fast enough to help our patients without all the side effects.
Great. And we have a couple of questions about the CD40 treatment that you had mentioned.
Okay.
One in particular with regards to when timing expectations for the Parker Institute study to open.
We hope this fall. We're working hard on that. It'll be a national study, and it'll probably take us the summer to finalize the plan. It'll come quickly, though.
Great. And then the other is regarding the administration and side effects of CD40. So the question is, how is CD40 administered, and what are the side effects?
At the moment, the clinical trials with CD40 have been to give the drug by vein as an infusion, just like the checkpoint blockade. There is some thinking to maybe try it either subcutaneously or even directly into the tumor. And there are clinical trials that are happening in Europe which are testing some of those ideas. We envision that using the drug IV is maybe the most practical and most straightforward. So far, the side effects from CD40 have been infusional reactions. So in a fraction of patients that get the drug, they can develop fevers and chills on the day that they get the drug. After that, it's actually very well tolerated. It turns out that the CD40 has become a very, very active area of investigation, and many companies and other entities are bringing forward different types of CD40 antibodies. And we're going to learn this year, I believe, whether and how each one of those drugs differs. And we anticipate that there will be different side effect profile with each one. Having learned from some of the earlier antibodies, there is a potential to generate a different type of antibody where those side effects on the day of infusion will be far less.
Great, thank you. And then we have a couple questions regarding the stroma of pancreatic cancer that you mentioned in your presentation. So the first question is, how does the stroma impact any immunotherapy approach in pancreatic cancer?
Well, that is being teased apart left and right. There's aspects about that which we're beginning to get hints about. The first is that within the stroma are a number of other cells in the body, immune cells and fibroblasts, which actually have very potent ability to neutralize T cells that come near that stroma. And some of those cells can create and produce molecules, cytokines, and other factors that further repel the T cells and sort of make it like the same end of a magnet trying to come together. Fortunately, a lot of those molecules are being understood. Many of them I referred to in the talk, and one by one we hope to derail them. Now, the stroma lately has become, because it's obviously so important in pancreatic cancer, the true role of the stroma is far too simplistic to say it does one thing or it does the other thing. As more experiments are done, we're learning many nuances, including some interesting immunological nuances. So I would say that research into this area is really a super high priority, and there's great effort in that regard.
Great, thank you. And then we have another question about the Moonshot initiative that you referred to. So how do you see the new Moonshot initiative tying in with the current research efforts of the NCI as it relates to immunotherapy?
Well, I think it's an accelerator. I think it's an opportunity and a challenge to all of us to do a couple of things. The first is to think bold. So we had, as I mentioned, the opportunity to hear the Vice President's vision on this, and he's looking for bold ideas. He's looking for us to say, we've come, we're on the inflection point. So instead of sitting back and saying that was good enough, how can we challenge ourselves clinically and scientifically to go to the next level? And of course, those types of high-risk projects are sometimes difficult to fund through conventional means. And so the promise is that the Moonshot is designed to really encourage investigators to bring out their boldest ideas. The second opportunity and challenge that is very clear from the National Moonshot effort is that all of us in this field, and that includes everybody affected by cancer, needs to do an even better job of talking to each other, communicating with each other, sharing the data, and think about how can we pull down barriers that prevent us from collaborating. One of the huge things from the Parker Institute was the pre-negotiated terms of collaboration, so those 6 organizations can talk freely amongst themselves and share data as it comes out in real time. So there's there are some cultural aspects of that which we have to address, but there's also, it turns out, there's also some technical aspects, bioinformatical aspects of sharing data, considering the size and the monumental size of some of these data components, that agreeing to share is one thing, but actually sending the information to each other is another thing, and that certainly is under the focus of the Moonshot as well.
Great, thank you. And it looks like we have time for one more question, and this actually relates to immunotherapy for neuroendocrine pancreatic cancer. So is there a actual or possible use for immune therapy in neuroendocrine pancreatic cancers?
Yeah, it's not my particular area of focus. It's a great question, and I appreciate it being brought up. One thing that we are learning is, initially we thought, we saw some of these successes with immune therapy and melanoma, and we thought maybe that was it. It would be melanoma, maybe just kidney cancer. It's just amazing as efforts continue to go forward. Every tumor type seems to have an immune vulnerability. We just have to be smart enough to figure out what it is. Amazing data at the recent cancer meetings about stomach cancer, liver cancer, small cell lung cancer, all being attacked by immune therapies. No reason a priori in my mind that neuroendocrine will be any different, and there are efforts in this regard.
Thank you. So we would like to again thank Dr. Vonderheide for his excellent presentation. The PowerPoint slides and a recording of this webinar will be available at pancan.org under the Educational Events page. There may be a delay of several days before the information is posted, and your patience is appreciated. Once again, I would like to thank our webinar sponsor, Lilly Oncology. A survey will pop up once you leave the session today. Please take a moment to share your feedback. If you have questions or would like a personalized clinical trial search, please contact Patient Central at 877-272-6226 and ask to speak with an associate. This concludes the webinar. Thank you for attending.